Great to see so many faces. This is awesome. Thank you,
Yeah.
folks.
Yes, thank you
Mhm.
all so much for being here today. I think in the interest of time, and with such an exciting audience already, I'm going to get started with some Bina announcements. Hello, if you are new to the world of Bina and our events, I'm Vanessa. I'm the Bina Program Administrator. So I'm just going to run through a couple quick things that we have going on as a way to share about what we've got happening with Bina, as well as give folks a little bit of time to enter the room. So with that, I wanted to share that we also have a scientific speaker series and a career speaker series. Those are on hiatus for the summer, but we're very excited to have a celebration of Hispanic Heritage Month starting on September 10th. That'll be every Tuesday throughout those months, except for when we're having our Congress, but I will let Nikki share about the Congress in a moment. And then our career speaker series, if you're interested in the wide variety of roles that imaging scientists can have, then please do join us for that series. We're here in June, so I just wanted to share this calendar. We're going to be on hiatus with most of our events throughout July and August for the summer because we know most folks are busy during that time frame. But I did want to share that we do have a user group that focuses around automating metadata, as well as a lattice light sheet group and an expansion user group. So we're very excited to kick off this fourth user group around STED. But if you use any of those other technologies, we encourage you to check out those sessions as well. And then if you happen to be in the realm of being a leader or a core director or manager, we do have leadership and management trainings. Those are full for this year, but we are starting our wait list already for 2025 because we've had such great interest. So if you're interested in those, please join us for that. And then we also have our Exchange of Experience virtual group on the last Wednesday of the month. This one will focus on translation efforts, but we've covered a wide variety of topics, usually loosely linked to some core facility management and tools and things that are beneficial there, but we encourage anyone to attend. And with that, I will pass it to Nikki Biley, our Bina Program Coordinator.
Hi, thanks, Vanessa. Thanks for sharing all of that great stuff that's ongoing all the time with Bina. This is our event happening in September. It will be virtual as well as offered in person. So please join us. Even if you are not able to come in person, we'd love to have you join for the virtual portion of it. It's a full week, essentially, of activities. Well, almost. Fridays are all optional activities. But it will have a great lineup of speakers. So please take a look at the webpage and join us. And I dropped the link in the chat too. And then the next slide is about some of our other activities available through Bina. So many of you may know about our professional development program, but we also have an exchange of experience program where you can have funding support to travel to a lab and learn a wide range of different skill sets, whether it be around STED or whether it be around an alternative technology, running a core facility, image analysis. There's a whole gambit of different opportunities there. So please take a look at that link tree, and I'll also drop the link in the chat for those opportunities, and reach out to us if you have questions about any of those. And if any of you run courses, consider submitting your courses for those PD programs. This is a quick thank you for our platinum sponsors, AVR Optics, Evident, Nikon, and Miltenyi Biotech. We really appreciate the support of our corporate sponsors. And I think the last slide just basically says, by joining this event, you don't automatically become a Bina member, and there are a number of benefits to actually becoming a member, not to mention the great newsletter that Vanessa sends out telling you about all the activities. So I've just dropped in the chat the benefits page where you can learn about some of those other things, which, as I say, includes being able to go visit other people's labs and have the travel and accommodation paid for and participate in some of the PD activities. So with that, I think I will hand it over to Ethan, who's going to take it away.
That was wonderful and effective, and I'm so happy to see so many participants come to this first meeting, this inaugural meeting of the STED user group. I have to say that in organizing and helping to organize this event, which is very well attended, I'm seeing, which is great. It's been an absolute pleasure to work with Bioimaging North America, especially Nikki and Vanessa. So I do want to thank them, or I do want to thank them for their help in making all of this run so smoothly. Before I get into introductions and we get into some of the discussion, just wanted to start with an inclusivity statement, which will be read at the beginning of every of these meetings. reminding everyone that this user group aims for an inclusive environment where all members of the scientific discipline feel welcome. We expect all conveners, presenters, and participants to honor our commitment to this shared goal. This includes offering mutual respect, regardless of career stage or identity. So with that, we can get into introductions. So my name is Ethan Peretz. I'm a postdoctoral fellow at UT Southwestern Medical Center here in Dallas, Texas, in the US. And I'm joined by all of the organizers who have helped really bring this to reality. So, you know, I'm going to start just briefly with what I'm hoping to use STED for. So that is, I'm using STED currently to try and image mitochondrial ER contact sites in the context of aging and neurodegenerative diseases. And so that's a little bit about myself, and maybe Madeline, you can next introduce yourself.
Hi, I'm really excited to see so many people here. I am an MD PhD student at UT Southwestern, also in Dallas, and I am finishing my PhD now. I've been studying mitochondria, particularly intermembrane proteins in the mitochondria and how they spatially organize and interact with other proteins, which are also in the inner membrane or on the outer membrane of mitochondria. So it's been really important for me to have like really good spatial resolution in my microscopy. I started working on STED for this project about 2 years ago, and went from working to STED first arrived at UT Southwestern. There were very few people working on STED, and so I was just really excited to get more opinions and see what other people are doing and how they're utilizing the technology.
Daniel?
Hi. Thanks. Oh, sorry. Hi, thanks, Ethan. So I'm a first-year PhD student, also at UT Southwestern. I'm currently working with Madeline, so also on mitochondria, like Madeline and Ethan, and yeah, we are trying to better resolve their ultrastructure. It's likely that I'm going to make much more use of the STED in the future, so having like meeting place where we can like share all of these ideas and hear different STED experts talk about like all of these amazing techniques is, I think, going to be very helpful for all of us.
Hey.
Hi, I'm Kate Luby Phelps. I recently retired from UT Southwestern like a week ago. And my job is pretty much to just learn things and stay out of the way.
Ina?
Hi, I'm Pina Colarusso. I'm the director of the Live Cell Imaging Lab at the University of Calgary. We're an imaging lab, and you'll see Dylan and Craig here today. There's a group of about 4 or 5 of us that support optical imaging with STED and super resolution. In general, topics of interest are mitochondrial biology, organelle contact sites, condensates, and the secretory pathway. The real reason I'm here, though, is that in the LCI and myself personally for the past 20 years is I see a lot of duplication and people trying to work things out on their own in their own labs. And I'm so excited to be part of this community because I think this is the best kind of learning when we exchange ideas and expertise freely and in a really supportive environment.
Guang?
Hi, my name is Guang Gao. I'm the imaging facility manager at the Life Science Institute at the University of British Columbia here in Vancouver, BC. Thank you.
Thomas?
Hi guys, I'm Thomas. I'm a professor at the Montreal Neurological Institute of McGill and the director of the microscopy core there. And I do love ultrastructure, and we have super resolution micro from very early on when they came into Canada, including STED. And I'm actually here to really help push the use of these techniques and democratize it more, and then help more people to use the power of these highly resolving microscopy techniques. In my own research, I'm a neurobiologist. I'm interested in synapses in the hypothalamus, and G protein coupled receptors as they work in neurons and neuroendocrine cells. And yeah, that's about that. So it's a little bit different from the cell biologists, but STED is great also in thick samples. And eventually we can also talk about that maybe in the group here. And I'll close with that, just saying today, because I have some family business, I'll be leaving the meeting very early. I just wanted to be here for the inauguration, but I have some stuff that I have to do that I can't put off. So I'll be gone after about half an hour. Sorry for that. Next time around, I'll stick around for everything.
Do we have Claire with us?
I am here. Thank you. So I'm Claire Brown. I am the director of the Advanced Bioimaging Facility at McGill University. And I was thinking I've been teaching about STED for more than a decade, but I finally got a microscope in our facility just last year. So I'm really excited to learn more about actually doing STED rather than teaching about it. And well, continuing to do both, I guess. But we're interested in focal adhesions, so looking at the substructure within adhesion contacts with the extracellular matrix. And I'm also really interested in pushing STED for live imaging, which is an expertise in our group. And I'm super happy to see this group of the next generation of imaging scientists taking a lead here. And so like Kate, I would be very happy to stay in the background, help you as you needed, but really let you take a lead. And I'm really happy to see you're all doing that. So thank you.
Nikki?
I'm sorry, I didn't expect to be. So I'm been a program coordinator, and I don't actually use microscopes, but I'm happy to support you all in whatever way we can with your microscopes and interacting and working together.
Vanessa, maybe remind people.
Yeah, hi everyone. I'm Vanessa. I'm the BINA program administrator. I'm based in Madison, Wisconsin. I did a PhD in chemistry, so I'm not myself a microscope user either, but have been supporting the BINA community for 3 years now, and it's just such a wonderful community, and I'm really excited to see you all here to focus on the technology of STED. So if we can support you in any way, please reach out to us. I'll put our email in the chat, and I think it'll be on slides in the future as well.
Just briefly, Vanessa, chemistry doesn't save you from being a good microscopist if you get the chance, right? There's many of them.
Yes, it is nice having that link.
Great. Thank you again so much to the organizing team for all of their efforts and input in really making this a reality and, you know, organizing. I see we're up to 45 participants, so that's really exciting. And so the open notes link has been dropped in the chat multiple times. And we're really hoping that everyone who's participating today can add their name as an attendee, so you can go and visit that, and we can have a list at the end of who was here, who's part of this community, and we hope that you'll be able in the chat to add a little bit so we can get to know each other better. So we really want this to be a community. We want this to be a research community where we're sharing resources, where we're sharing our science. And in order to do that, I think it's important that we actually have a chance to try and get to know each other. So if you don't mind going into the chat and just, you know, putting a few words or a sentence about what you hope to image with STED. And then again, in the, you know, hope that we can make this a really sharing, collaborative community. We hope that some of you might even, you know, raise your hand now, and we can discuss various aspects of what we want this user group to be, how we want it to serve this community. We're very interested in this being a crowdsourced community of ideas that really serves those who are attending this meeting, and starting off with what kinds of outputs that members of this community might want the STED user group to produce. So I hope that some of you can overcome, you know, some initial hesitation to speak up. And, you know, in really the hopes of, you know, sharing that this is a group of researchers whose experience with STED really spans a large array of experience levels, all the way from those who have never used STED before to those who consider themselves expert users. And I think that's really important and should make everybody feel comfortable in sharing, you know, their hopes and dreams about what this community can do for them. And then maybe before we jump into that, so hopefully some of you will start raising your hand and we can have a discussion. We'd like to just encourage you either in the chat, or you can email me personally with CC to Bioimaging North America. If we do want to include as part of this this meeting, each of these meetings, which is going to happen once every 2 months, a presenter who would be willing to share their research for about a 15 or 20-minute presentation as we're having today. And so if you or someone you know you think would be well suited to giving such a presentation, please don't hesitate to, you know, drop that in the chat now or contact me. My email is listed there. So I'm hoping with that, I don't see anybody raising their hands. So this is an icebreaker, everybody. So let's try to break that ice.
We do have a few people giving some comments in the chat. Maybe we could kind of look at those too.
Sure. Do you want to highlight anything, Madeline?
Sure. I'm seeing like a few people have agreed at the beginning that they're really just hoping to be able to help their core users. I am curious if you guys could respond on that. Are these like the STED is available at a core at your university and people can submit samples to have imaged? I think, yeah, like I know that Ethan and I, our experience right now is definitely doing it like mostly ourselves. So I'd be very curious to kind of hear what people's infrastructure is like at their university for this or at their institution. I'm also seeing a lot of people mention live imaging, and I know that's something that we discussed when we brought this user group together, that because STED is so powerful, we've had a lot of issues with live imaging being quite damaging to cells. So that's very much something that we want to talk about. I'm seeing a lot of people talking about mitochondria and neuronal structures. Definitely things that we have had experience with and would like to talk more about too. Some people are just excited to learn about new techniques, resolving ultrastructure. And someone said, wait, you let your users touch the microscope, which is hilarious. Yes, spent a lot of time in that room touching that microscope.
We have a few hands. I should also thank Madeline. She's also contributing to taking notes. So thanks a lot, Madeline. And those notes, again, will be accessible to participants after the meeting to revisit. So, Thomas.
Thank you, Ethan. I just a little shout out to the core facility people, obviously, who made the comment they just want to image whatever their users bring to them, which is also great. That's our job. But in my experience, I had the STED microscope very early on. We have it for 8 years now, and it's a new technique. And to the institutions where you bring in a new one, it is still new. So you will have to nudge your users a little bit, like advertise it and do some things that you can do on it and show what the power of the thing is, because otherwise it'll take a long time to catch on and seeing more use, at least in my experience. And I'm sure Claire has a comment for that as well. And I don't want to say more because, like Claire, I want to stay in the background here, but that I would want to shout out to those people. Thank you, guys.
Claire.
So we are at least starting with what we call assisted acquisition, where the users bring their samples after consultation and sample prep, and they work with our imaging facility staff one-on-one. And then we're thinking, you know, eventually we will identify users who are using it enough or seem to be really catching on that will train and allow it to be independent. So right now we're only doing it with assisted acquisition. And I just wanted to comment on what the group can bring. I think we've been even struggling just with how to set things up. Like, you know, you're not used to making your pixels so small. We're always trying to keep them larger. And so a lot of those things are like how to set up your scan speed and your pixel size and analyze your images and all of that, I think would be really useful if this group could gather and share best practices and protocols, benchmarking the instrument, those sorts of things. So.
You know, I should mention that some of you are already doing this, which is quite marvelous. But if you feel comfortable turning your camera on, you know, that's always one big contribution to a community and making people feel comfortable. And so I encourage you to do that if you feel comfortable and so inclined. So thank you. Katerina?
Hi, I'm Kate, actually. That's much easier. So I work at a microscopy core at Mount Sinai in New York. And I just wanted to add that it's exciting because we don't want to only, like, I hope we're not going to only talk about imaging, but also about sample prep, new, I don't know, chromophores, about new analysis techniques. So obviously everything addition to the imaging, because we mostly don't image for our users. We try to train them as much as we can so they are very independent. But we're also trying to help, obviously, to find the best staining techniques or best analysis and everything else that is around the imaging.
Fantastic. Yeah, that was exactly one of the aims that the organizing / advisory committee had talked about prior to this meeting, as could be a major output that could be extremely useful to this community. So thank you for that comment.
I think creating a bit of like a place that people can go to where we understand better, like the little bits of like, I feel like scientific lore essentially that are associated with doing a new technique, where you have things that like work for someone and aren't necessarily a part of like the technique or the methods that come from the company when you order like a dye. But that turn out to be quite true. I know something we've really experienced at UT Southwestern is the difference in cell type and how a lot of these live cell dyes work differently in different cell types. So we've been trying to get U2OS cells to work, and it appears that most of these dyes end up pretty toxic in U2OS cells. So I would love to just be able to, I think, have some kind. We discussed like having a poll or something where people could say like, this is a dye that I've used. This is how it worked for me. You know, thumbs up or thumbs down, and then just being able to access that repository to know if someone else has tried it, because it's such a hard thing to get a technique like this going when there's no one really to ask, because there aren't very many people doing it. So I think that would be something we could really create, like a database of this information.
Hi, I'm Damien. I work on the internal core facility at Janelia, and we have a Bayer STED. We had it for almost about 2 years now, and just as Claire was saying, we had to handle it a bit differently from other microscopes. We've started to do a lot more what you call like assisted acquisitions. We don't necessarily train directly people on the system. We first discuss their experiments. We try it with them. We see what they can get out of it, and only then we start training them really, because there is also a lot to cover to make it work. I would say. And also very often we try to give a little bit more support than usual to make sure it works in their hands with their samples, because it's very easy for the experiment not to work and just to go away from a microscope. So that's some just experience.
Thanks so much for that comment. Cibredo, I don't know the first name.
Craig.
Oh, Craig. Hi, nice to meet you.
So Pina and I had actually just recently written a blog post on 2-photon fluorophores. And one of the things that struck me in hearing the discussion now that paralleled what we discovered when kind of looking through fluorophores for 2-photon is that it's pretty easy to find kind of the optical parameters of a dye, but finding kind of the biophysical information about the dye, that learned experience, is actually very difficult. And I'd love to see some kind of structure, whether it's something that gets added to fpbase.org or one of the other big repositories, like little notes, sort of, if you're using this for live versus fixed versus STED, or even any of the other sort of unusual, like Palm, Storm, all those super resolution effects that are kind of frankly hard on the dies. But it would be great to see like that kind of information included in either those databases or a new one.
Absolutely. Thank you so much. That's also a key aim, I think, of this community, right, is to get us all in a room where we can share exactly that experience. This, you know, I don't even know how many years one could say is built up of experience in this Zoom call right now. It's very exciting, and we should leverage that as a key resource. So thank you. Also, you know, as there's a little pause in the activity of people raising their hands at this moment, this is the first meeting. We want to keep growing this. We've leveraged our own networks in order to encourage people to come and join. You know, one thing that we could do as a community to keep growing is if all of you go out and leverage your networks to invite more people who you think may be of interest to joining this working group that's going to be meeting again once every 2 months. So. Well, if there's no more comments.
Can I make one more comment? I was just going to put a long note in the chat, and I thought it might be easier to just say. I was wondering maybe we could make a rough list of good and poor STED dyes, and maybe put a little article in Focal Plane announcing this user group and offering that as like a first resource, even if it is just a first pass and not necessarily comprehensive. But just a thought.
If I may add, that was something I was thinking about too, but as I wrote initially in the chat, unfortunately, I'll have to quit at this point today. I promise next meeting I'll be around as a resource or whatever, but unfortunately now I have to leave you guys. So have fun for the rest of the meeting, okay?
Andréan.
Hi, I'm Adrienne. I'm in Flavie Labocardinal's lab here at Cerveau in Quebec. We own an ABR expert line for a while, and Flavie just got a nice grant to start our own platform for super resolution microscopy here. And I really like this idea of this database of good or bad, but in my experience, it's been so variable of what is good for one experiment or bad for another that a list might be a bit reductive. And so, for example, we've had success doing STED and Storm dyes in really specific conditions for phlegm or whatever. So it's kind of hard to say it's good, it's bad, but it would require more explanation. But I'm not sure which format would kind of accommodate for that.
If I might comment on that, one thing I noticed when going through FP base that did save me is they're very good about linking to the original papers where the dye was first explored. So yes, you're absolutely right. That kind of background information would be mandatory.
Awesome. Thanks for the comments. So I think this is probably a good time to transition from you know, this more discussion part of the meeting to our presenter. You know, going once, going twice. If anybody has some last comments, of course you can keep posting in the chat throughout. If nobody else wants to raise their hand, I'll just start by introducing our speaker for today. So Guang is the imaging facility manager from the Life Sciences Institute Imaging Core Facility at University of British Columbia. He completed his PhD in cell and developmental biology at the University of British Columbia under the supervision of Dr. Ivan Robert Nabi, where his dissertation was on endoplasmic reticulum, nanodomain organization, and communication with mitochondria. I'll highlight that Guang has a recent publication in Journal of Cell Biology on membrane contact site detection, especially mitochondrial ER contact sites, and maybe we'll hear a bit about that today. Guang has also received honors and awards throughout his career, including a Faculty of Medicine Graduate Award and the Dr. Laura G. Jash Memorial Prize from University of British Columbia, which was awarded to a student supervised by a faculty member of Department of Cellular and Physiological Sciences, exhibiting the best combination of research achievement and teaching or service. And yes, Guang has graciously accepted being our first presenter in continuation and continuing demonstration of his service to the community. So thanks so much, Guang, and take it away.
Okay, all right, thank you. First of all, I'd like to thank Claire, Pina, Thomas, Kate, and Nikki, and Vanessa for initializing the conversation, set up this user group. And thanks to Ethan for the introduction. Today, I'm going to talk about STED and its applications. Okay. So at the beginning, I just want to see some images or diagrams might have been adapted from some commercial companies, but I have absolutely no commercial interest with them, just to make it clear. Okay. So to start with, the signal pathways events at a molecular level, subcellular, cellular level, control or regulate the development of human body, for example, aging. But however, this is not always the case, as disorder occur. When this happens, very often we need to go back to the subcellular level, molecular level, or cellular level to find the pathological causes, to correct them, to target them. And imaging is one of the very important research tools we use nowadays. For example, wide fields will allow us to see the spatial distribution of different population of cells within the tissue. Very often we use confocal to identify, to localize the virus marker or different markers within the cells to subpopulation, cellular levels. For example, this is a Calo3 cell infected with SARS-CoV-2 and labeled with two markers, double-stranded RNA and a nucleic capsid as a viral marker and two host marker, DIP and actin. With super resolution, we can even look into the cells and at the subcellular level to see all the details necessary for us to understand the biological mechanism. For example, this is the endoplasmic reticulum 3D rendering, where we can see the nuclear loops in the middle and all the details in the periphery of the cell. With the super resolution, let me see, I think. With the super resolution, single molecule localization, and we can even look closer at the cell, at the big protein complex to the molecular levels. Okay. All right. So what do we have in the toolbox in terms of super resolution? Here is a summary of several of them. First is a single molecule localization, and we have a STED, and the same is not listed here, but we have expansion. And also we have MinFlex for 3D, 2D, for fixed samples, and also for live cell tracking. Of course, today's focus is STED. And then why we would need a STED? And there's a limitation. Excuse me, but I think something is blocking a part of the presentation. I don't know whether you can see them, all of them properly. Let me see.
We can still see fine.
Okay,
it's
perfect.
Not covering. Yep.
Okay, perfect. Thank you. Yeah. So the limitation is we cannot guide the light, the laser, to a perfectly focal panel, but rather it is very often is area which is very large, a round area. The area is the size is defined by IB light, IB's diffusion limit, and which is determined by the length of excitation and also the NA, And in practice, this is around 200 nanometer. Then the solution instead is control the size of illumination, not only make them dimmer, but rather just to only focus on the central region of illumination. And the magic is with the donut. How it works? Then for this, we need to go back to the some diagram where we have the excitation and this reaches the sample, and then very quickly we have the emission from the sample. Well, the spontaneous emission doesn't have to happen as it can be controlled if we have a redshift light, and with the photon which reach the sample right during the emission, and which can convert the emission into something into a wavelength which is same as the redshift light. That is called stimulated emission. If we apply a donut shape of redshift light to the sample, and then the light of the which is called STED beam can convert the spontaneous emission into stimulated emission, and consequently we have a switch. So the switch is the peripheral emission now is switched into spontaneous emission, while the middle is unchanged and it remains as spontaneous emission. And by setting up the detection window, which is right between the excitation beam and STED beam, then we can perfectly detect the central region of the emission. And further, this size of the emission is now be redefined, and which is determined by Imax STED, which is the maximal intensity of the STED laser used, and also the RST, which is half the intensity, the property of the fluorescence emission get reduced to half. And further, the size of this emission is controllable because we can increase or decrease the STED laser and also the excitation laser to determine the size. And by using continuous waves excitation or pulse laser, now we can see clearly what is in the middle of this big structure, the big illuminated area. Okay, now I have some examples to show you, and which will be focused on one structure, which is endoplasmic reticulum, ER. So the ER was first reported back in 1954 by using electron microscopy. And the ER is a very convoluted membrane structure and carries multiple functions, including lipid synthesis, calcium store, membrane contact, and unfolded protein response. And it also provides the site for protein synthesis and involved in mitochondrial dynamic, obesity, diabetes. Structurally, ER can be divided into 3 morphological structures: and peripheral ER sheets, central ER sheets-like structure, or peripheral ER tube structure. And STED on most the super resolution or mostly STED has allowed us to revisit all those 3 type structures. For example, in central ER sheets-like structure, in the past, it was intensively studied in yeast or in tissue by electron microscopy, and the 3D reconstruction and shows the ER sheets-like structure. But now we can see the similar structure by using STED. And this is HT anti-cell. You can see in the central region, we can see the ER sheets-like structure, as we can see with the electron microscopy. And for the peripheral ER sheets, so the peripheral ER sheets, as you can see here, labeled with DL, which is the diffusion limited imaging technique, and label shows them as a peripheral ER sheets-like structure. But by comparison, you can see in STED and 3D, in 3D sim, and they are like a very firmly packed tubular structure. It is termed as ER matrix. And similarly, we can see this as those structures with STED. And you can see there are nanoholes in the peripheral tubular structure, and appeared as a so-called ER matrix. Then how about the tubular structure? So tubular structure, if we draw a line, and for the tubular structure across them, and for confocal and STED, and measure the fall width at half maximal. Confocal measures the size to 50 nanometer-ish, and by contrast, the STED gives us around 6 nanometer for the weight, which is almost the same as what is measured by electron microscopy. And more interestingly, if we look at the structure along the tubular, and you could see with confocal, it looks like a solid line, but by contrast, STED allows us to see all the nanodomains along the tubular structure. The size of those nanodomains are controlled by two shaping protein, Klimt-3 and reticulum. And similarly, a different approach and showing the same phenomena as you could see here, as indicated by the orange arrow, and they call them very vertical sites on the ER. Okay, and hopefully I can convinced you. STED is doing pretty good in terms of spatial resolution, but how about temporally in live cell? Here is one example where, and using STED, and we can see all the nanoholes in the peripheral ER sheets. Those sheets abundance of the holes, the abundance are controlled by two shaping protein. One is reticulum, and the other one is Klimt-3. And similarly, we showed with the live cell, we can track the dynamic of the ER nanodomains along the ER tubular structure. And when we track them, and we found those ER nanodomains are very dynamic. But when they travel around, they are limited to a certain range of the ER structure. And the dynamic of the range or dynamic are controlled by, again, two shaping protein, Klimt-3 and reticulum. And of course, we can do two color can be, live cell can be done. And this is an example where two marker 661 and KDL labeled for different components of the ER. So these are two color live cell. And similarly, we can do live cell in 3D if the acquisition, if we focus on a smaller region rather than a big, the entire cell. Similarly, we can do multiple color in 3D. Here is an example where two structures are labeled. One is ER in green, one is mitochondria in red, and imaged from top to bottom to get the idea of the ER mitochondrial contact in 3D. And then we also developed a method called the contact site, MCS contact. And as you can see here, and we detect the contact globally inside the cell, but not only restricted to the peripheral or the central region of the cell. Okay, and with that, and I would like to thank, first of all, I would like to thank Dr. Robert Nabi, and he is the director of our imaging facility manager, and he put a lot of effort to develop and to grow the imaging facility by writing different grants, bring in new instruments. And I would also like to thank other faculty members at the UBC who contribute different instruments to the facility. And at the end, and I will also personally thank Dr. Fan Rinman, and he is now with Avangent. And in the earlier years of my PhD, and I learned a lot from Ray and in terms of the confocal, and I still remember the time he trained me on our very old FE1000 And also thanks to the funding agency. And I would like to answer any questions you could have. Thank you.
Thank you very much. We have time for questions. If there were any images that you saw and you're wondering, how do you do that? You know, this would be great.
So what state platform have been used for this study and how were the images processed? Are these raw data or processed in certain ways?
So the platform we have within our facility is a Leica SP8 3X STED. It came with 3 depilation lasers, STED beams, and 592, 660, and 775. It was installed 10 years ago. So some of the images are raw image, as you could hear. This is but the rest are deconvoluted or rendered with, let's say, MRS in this example, and where it was first deconvoluted and then we rendered the image with MRS. Yeah, deconvolution is only thing we do in terms of processing.
Do you have a particular deconvolution module for Ameris specific to STED? And if so, how do you feed your point spread functions into that thing?
Yeah, this is a good question. So well, as we all know, at the very beginning of Leica STED development, when they sell Leica SP8 confocal, it comes with a package of a Huygens deconvolution package. And we have been used the deconvolution package since then. Later on, Leica comes with its own like a lightning. But we decide by side comparison, we didn't see a dramatic difference. But to be consistent with our earlier study, we just decided to go with Huygens Pro deconvolution. Yeah.
Paul?
Great talk, Guang. Thanks so much for that. I was just curious a little bit on the tracking that you did of the ER nano domains. So was this a manual tracking? Was this automated? Was this in Imaris? Can you just comment a little bit on how you did that?
Yes. So this one is program we use called ICE. We didn't do we didn't use the Imaris program at this time. We don't have the Imaris yet, but rather we use a program called ICE to track the dynamic of the nanodomains along the ER structure. I can type the name of the program called ICY. Any other questions?
Are there any kind of overarching tips you have for live STED? Like some of the things you have to maybe compromise in order to keep the toxicity levels low?
Yeah, I think several things we did. So first thing is we try to image the cells within a very short period and try to go very fast with the image acquisition. This is we tried. For example, this one is 25 frames per second, and then it was finished within 4 seconds, with a total of 100 frames. And after that, we saw if you go longer, I think the phototoxicity is just too much. And as we all know, the ER is such a fragile structure. The structure can get damaged if they are not happy, and the structure get changed dramatically if they are not happy.
So do you sacrifice resolution to go faster? Do you make your pixel size bigger? How do you do that?
I didn't really go to the pixel size too large, because if we go to pixel size too large, potentially we will lose our resolution by doing that like Nyquist. But rather we decided to go with a very small ROI, and first would allow us to increase the imaging speed, because the ER itself is moving so fast. Actually, for this project, at the very beginning, we tried different speed, and then we realized the lower speed cannot catch up the dynamic of the ER nano domains, and we had to go to very fast. Yeah.
I wanted to ask, so when you use the convolution, is it mostly to improve your to improve further your resolution, or to reduce on depletion power? That's one question. And the other one, I was wondering what was your Z resolution in these experiments?
Okay, yeah, that's a good question. So for the first part, for interim deconvolution, you know, the Huygens professional, they have different, like for example, single node ratio, quality thread, quality control. They have different controls for you to similarly or genuinely for you to do the deconvolution. And very often we go different signal ratio and other numbers and compare them. For example, signal ratio, we start from 3, 5, 7, and compare them side by side with the raw image, and then we determine which one match best with the raw image. So sometimes maybe in the raw image, the phenomena is not that obvious, and the deconvolution could slightly help us to highlight the phenomena that wasn't that obvious with the raw image. But we don't use deconvolution to purely increase contrast, but rather to slightly reduce the noise, reduce the noise in the background. So what was the second part of your question?
Oh, it was just was wondering what was your Z resolution.
This is a good point. We didn't really get a base measure Z resolution, but rather we rely on several things. The first is based on our understanding of the N plus reticulum, for example, and then we optimize the how much static power we can borrow from the XY and apply to Z to see because the ER is a tubular structure, and we want to see the ER structure as close to a tubular structure as possible in the Z. This is the criteria we use to optimize how much power we borrow from the XY.
Okay, I see. Thank you very much. Thank you.
Okay, thank you. I see that we're entering the final minutes of the meeting. I myself am being kicked out of this seminar room that I'm taking the call from. But I do want to mention before we wrap up, once again, thank you all for coming. Thank you, Guang, for the great presentation. Thank you to BINA and the organization and all of the effort done by BINA and the organization and members of the advisory group. our next meeting will be August 8th, again on Zoom. I've also dropped, a reminder that in the chat to email me if you or someone you know would like to present at this working group in the future. That would be great. And I do want to thank everybody for coming, everybody from academia, from industry, if they're here. So thank you so much for making this, what looks like it's going to be an exciting and thriving and collaborative community. So thank you so much.
Can I just jump in the last couple seconds? I just wanted to say that the chat is going to be saved, and I think we can share it in the same area on Google Drive where we have the STED user group. The recording is going to be posted on YouTube. As far as going forward, I personally would suggest that we alternate between presentations by specific people like we've had today and topical discussions on things that bother us, that we need answers to make this practical. And so it seems to me that we could have a document in the STED user group area where people could put down the topics that they're interested in discussing, and then maybe in August we should have a discussion on one of those topics. I've seen some of that coming up in the chat. So I'm not running this thing, but that's just my 2 cents.
Great. I see some thumbs up there. So thank you, Kate. Thank you again, everybody, for joining us, and we hope to see you next time.
All right. Thanks for hosting.
Thanks all so much.
Yeah, thank you everyone who made this possible. It's exciting.
Thanks. Bye.
Congrats. It was a terrific turnout. Thank you.